field for more than 20 years, and if you've been here long enough, you give them some license for self-indulgent behavior and come up with some cheesy titles and pithy observations, but I think they contain some general truth. And as we all know, wisdom is acquired through the use of maudlin aphorisms and wise quotes from famous people. So there will be 3 parts of this talk, each one 15 minutes long. And first, everything should be made as simple as possible, but not any simpler. T-cell therapy is very complicated. And certainly with all the tools we have available now to study and manipulate T cells, even more so. And then secondly, what's old is new again. And then finally, the power of one. So let's just jump right in here. And I think that the first quote by Albert Einstein applies very well to immunology and T cell immunotherapy. The inside joke is that wherever 3 immunologists are gathered, there will be 6 opposing and contradictory opinions. So to help us illustrate some of this in a more simplistic manner, Anke Verban is a visiting graduate student from Austria who produced some of these images. And the first thing you need to know in T-cell therapy is how T cells divide, because a single T cell cannot do it alone. T cells obviously will proliferate and expand. They have a finite doubling efficiency. And to induce that doubling, maintain persistence is critical for T cell therapy efficacy. And second, obviously, is making contact with a target. T cells must then make contact via an immunologic synapse, or a SMAC, supramolecular activation cluster, which involves interaction between the peptide MHC complex that's found in tumor cells and the T cell receptor, as well as accessory molecules on both sides, leading to what we see here is a small clip that Anke made. So the ghastly display of violent tumoricide, where a tumor cell is besought upon by one or more T cells. You see a lot of blebbing and evisceration in the final moments of this T cell here. Turns from green to blue. You can see the nucleoli actually condense. Here, 2 of the T cells, we don't know if they're acting on the same target or not, but you see the binding and you see death. Not sure why this skull shape is here, but that pretty much is a dead tumor cell at this point. And they'll go on, recharge itself, and kill the next set of tumors. So one of the first things, though, that is, I guess, fundamental to T cell recognition is how a T cell recognizes its target. And it does this because the tumor antigen from any compartment in the cell for T cell receptor-based killing, this tumor antigen is processed and presented as small peptides on the surface in the context of self-MHC. And when this happens, the T cell receptor has an opportunity to see that particular fragment, that small peptide fragment that's brought to the surface. And this is what a naturally occurring T cell see, or a TCR-T engineered T cell. So keeping in mind again that that antigen or that protein can come from any compartment in the cell, including the surface, of course, but also transcription factors and so on. Whereas a CAR T cell recognizes only the surface antigen through a chimeric antigen receptor, which is a hybrid of an antibody, or scFv, some part of the antibody, to epitopes on the surface of a whole tumor antigen. Now, there are exceptions. There are T cell mimic antibodies, and we can talk about that later, but by and large, CAR T cell therapy is based upon recognition only of surface proteins. The most important thing, obviously, in our discussion is how to make T cells. And there are really 3 basic modalities, and I'm going to use some Olympic memes just to convey some of this across. And the first of this is the TIL, or tumor infiltrating lymphocyte. Strategy was pioneered by Steve Rosenberg back in the 70s and 80s, and really now has achieved FDA-approved status in the form of a number of cell therapy companies, which are now using this in clinic for treatment of patients with melanoma and possibly other solid tumors, but involves isolating T cells from a T cell rich tumor biopsy sample, expanding them usually with high dose IL-2 ex vivo, and giving that back to patients. And so this is really the OG of cell-based therapies, and I think that's well represented here. I think the other 2 now that are discussed in this presentation are TCR and CAR T cell based therapies and ETC therapy, both of which are considered antigen-specific based approaches. CAR T cell, TCR-T immunotherapy, we're all familiar with, here represented by a highly sophisticated, technologically endowed air pistol guy, because these T cells can be genetically or CRISPR modified for tool targeting, to armor them, add switch receptors, and to knock out and knock in various genes. Endogenous T cell therapy, on the other hand, uses natural occurring T cells, which are sourced from peripheral blood, and probably something I identify more closely with this Turkish individual, not because of his dad bod, but because he does resemble the unconventional nature of our approach, using naturally occurring T cells with intact circuitry, intact external cues, and allowing for epigenetic control of T cell fate and differentiation. So this is the unconventional wisdom guy. And I think the first sort of blanket statement, which has been pretty much held in belief over the last 20 years, is that there are really no tumor-reactive T cells in the peripheral blood. And these can primarily be found where the action is, in the TIL or tumor site, or you have to engineer the CAR T TCR-T receptor. And certainly more than, I guess, 20 years ago in the 1990s and early 2000s, any suggestion that these T cells could be found in peripheral blood was heresy. It was pretty outlandish, but at that time, Mark Davis and John Altman had invented a method for identifying T cells on a structural basis alone, and that is the peptide image C tetramer, which is the natural ligand for the T cell receptor. It's a multimerized construct of peptide image C that T cell recognizes. And we found, as you can see here in this paper in Nature Medicine, and subsequently in Genomic Immunology, that there are circulating populations of antigen-specific T cells that are present, which are potentially tumor reactive. And these are MART1 specific, GP1 specific, and tyrosine specific. Very unusual and very, very low frequency because these recognize normal self non-mutated proteins. However, we went ahead anyway and isolated and cloned these. Sometimes we would have 100, 200 plates, so 10,000 clones that we had to sift through and identify and then expand. And as it turns out, these were in fact tumor reactive, recognizing tumors in an antigen-specific fashion. Not only CD8 T cells, but also CD4 T cells against NY-ESO-1, for example, when the class 2 restricted epitope was identified. So now these T cell clones recognize melanocyte-associated proteins and could be infused into patients to mediate some sort of clinical efficacy. I think really this is taken down. But the next sort of barrier that faced the endogenous T cell therapy world was sort of a blanket statement that T cell clones don't work. And this is an actual quote. I'll explain that a little bit more later. But when we had the opportunity to isolate and clone these antigen-specific T cells and also expand them using a rapid expansion protocol, which was invented by Phil Greenberg and Stan Riddell, we were able to treat patients, in this case, with advanced disease. This was before the time of checkpoint therapy, and so there was only high-dose IL-2, perhaps TIL, interferon, and so on. We had grown about 10 billion of these T cells that recognize MART1 for this patient who, as you can see here, has a metastatic lesion hanging off his transverse colon. And not only did these T cells persist in vivo here up to 508 days with fairly high frequency, 1.5% of all peripheral CD8 T cells, but they were able to mediate complete tumor eradication. This patient is now 12 years from treatment, high school teacher in Seattle, now retired and alive and well. This was published a few years ago, quite a few years ago, but I think the conventional wisdom at the time was that really if you didn't have TIL as a source of T cells, this wouldn't work. And I think that at that time I was at Fred Hutchinson. And it was quite interesting because I was junior faculty there. When this was published, Patrick Crew here at MD Anderson said some very kind things at the time about the research, which was really unconventional. If you didn't use TIL, you were probably marginalized in the research community. It was also the first time I encountered bullying in the academic world, which, by the way, should not be tolerated. And I guess Steve Rosenberg felt it was important to point out that clones do not work. The basis of which was unclear to me and anyone who read the paper. But I guess if you say something enough times, people would believe it to be true. And so we face a lot of opposition in trying to move this further into clinic. Not sure why I said that, because if you do the math, it's pretty clear that there's a significant opportunity for T cells to proliferate, whether they're clones or not. So for a single cell to expand to 10 billion, it requires 34 doublings. So pretty simple math. And if you take cells early enough, because there are 50 to 70 doublings in the T cell lifetime, and probably even more, you certainly can understand why this process can lead to an additional 5 doublings, a gigantic lymph node. Basically, you replace all the T cells in your body if there were 5 more doublings in vivo. And certainly don't want that to happen, but it certainly reflects the potential or the capacity of T cells, whether they're clones or not. But it does mean that It depends on where your starting population is. Your starting population is at the terminal effector stage. You certainly don't have too many doublings left for these T cells. And if it was earlier on, such as a central memory T cell, there's a huge capacity for these cells to expand after adoptive transfer, as was later showed in a number of clinical trials. Which leads us to this question, really, all T cells are equal, but are they? But some are more equal than others. And apologies to George Orwell for abstracting this from Animal Health Farm, but I think that this speaks a lot to the identification of what's considered a good T cell for adoptive cell therapy. And so when we did this study, where we had sort of stochastically identified antigen-specific T cell clones from patients and infused them, we found that patients who had progressive disease had very short-lived T cells, as you can see here, and not surviving in the blood more than 1 to 2 weeks, whereas those who had complete response had long-term persistence of these transfer T cells. And we looked at the phenotype of these cells. We find it went in looking as effector cells, the CD45 RO positive, CD28 negative, CD127 low. CD127, by the way, is also the IL-7 receptor. So these were truly effector cells going into the patient. But what came out of them in the case of progressive disease was no change in their phenotype. But in the case of those who had persistent transfer T cells, upregulation of CD28 and CD127. So coming in, they all looked the same, but certainly they were more equal in the case of the patient who had a complete response. And what we're looking at, in fact, was persistence of a transferred central memory type T cells as opposed to an effector or terminally differentiated cell in these patients. Effector cells of reduced capacity do not persist long-term and fail to eradicate tumor, where central memory cells persist, as in this case, almost a year, and continue to display central memory and actually recover the central memory phenotype. So it's really not what happens on the surface that's important, but perhaps inside, in which cells which have a high proliferative capacity and the potential for effective function can be isolated and expanded. So it turns out that to acquire this understanding of what a more equal T cell would be, you cannot Frankenstein in an antigen receptor, disrupting the natural circuitry in T cells that have evolved over millions of years. You need a naturally occurring T cell with intact circuitry, receiving intact external cues to show the way. And believe that insight really was achieved because we were able to do studies such as this with endogenous T cells. So among all the gamma chain receptor cytokines, it turns out that IL-21 is unique. It's a key driver in pushing naive T cells towards priming into a central memory T cell. And you can see that in contrast to the other gamma chain receptor cytokines, IL-21 leads to enhanced expression of CD28, a regulation of CD27 and CD127, and functionally enhanced proliferative capacity and also a higher energetic and metabolic profile. And so with all this in mind, we were able to move to additional first-in-human trials where we prime naive T cells in the endogenous T cell therapy protocol to become central memory cells. One fairly important study that was published in Science Translation Medicine actually took advantage of this sort of dichotomy at the time, in which initially WT1 specific T cells generated without this IL-21 priming protocol, because that was the standard at the time. And when we treated these patients with high-risk leukemia after transplant, it turns out that while you can infuse WT1 specific T cells to recognize WT1 positive AML, they did not persist. And unfortunately, in all these patients, the leukemia came back. You can see the leukemic fraction reaching 100%. All these patients relapsed, unfortunately, passed. Whereas using the central memory protocol, we generated central memory type T cells, and even after last infusion, these T cells persisted for more than a year. And none of the leukemia came back in any of these patients with obviously minimal residual disease, but high risk for leukemic relapse. We took this further to solid tumors by combining that with anti-CTLA-4, as you can see here. At that time, anti-CTLA-4 had just been approved, and we were able to add this to the regimen immediately after T-cell infusion. So in this particular case, patients who had failed anti-CTLA-4 therapy were treated with IL-21 prime T cells in combination with anti-CTLA-4. And not only small volume, but also large volume disease was eliminated. Again, this patient is a long-term survivor whose T cells still persist in the peripheral blood years later. No high-dose lymphodepletion, no high-dose IL-2. And so if you take a look at this sort of protocol we established in a fairly empiric fashion, the question then was, what was the mechanism of IL-21 priming? How did it go about generating this population of central memory T cells? And it turns out that unlike the other gamma chain receptor cytokines, IL-21 drives the signal primarily through STAT3. So IL-21-driven phospho-STAT3 binds to the promoter region of important or I guess well-established prototypic central memory type cells, such as memory type genes such as CD28 and drives its expression, and certainly leads to sort of the upregulation in both CD28 transcription, as you can see here with the shRNA. Certainly, the only way to prove this was to use knockout humans, and we didn't have knockout humans, but fortunately, Dean Lee provided us with patients who had Job syndrome. And if you know patients with Job syndrome, the genetic defect is a loss or essentially an autosomal dominantly functionally negative STAT3, and these patients could not regulate CD28 even when the T cells were exposed to IL-21, proving in fact that this was a direct mechanism of action. And if you take this a step further, you ask the question, well, if it works in naive T cells, why can't it work in TCR-T and CAR T cells? Unfortunately, if you expose these cells or these cell products, which are composed primarily of effector cells, although there are some memory and naive cells present, for the most part, these effector cells do not respond to IL-21 priming. Phospho-STAT3, which is driven by IL-21, just bounces off the promoter region of CD28 here because of the histones. And so the obvious solution was to pre-treat these T cells with an HDAC inhibitor, which we did. And if you do that, you now expose the region that phospho-STAT3 can bind to, and you can generate these central memory type T cells and sort of what we call epigenetic reprogramming, both phenotypically as well as transcriptionally. These cells appear and function through this process of HDAC inhibitor exposure followed by IL-21 priming to behave as central memory type T cells. And now we can now apply this tool not just to endogenous T cell therapy, but all the other modalities as well. And I think these are sort of insights you acquire in part because of the sort of systematic approach to understanding the basic biology of T cell memory and differentiation. And I would say this statement is pretty much bulletproof. So I think because conventional T cell therapy at the time, TIL, CAR-T, and TCR-T, have generally required conditioning lymphodepletion to achieve T cell engraftment and persistence in vivo because most of these cells are not memory cells. This regimen of cytoxin fludarabine has been applied and has been very successful, obviously, in ensuring that these T cells engraft and persist. But it is relatively empiric. In fact, the 5+2 regimen is empiric and based on non-myeloablative stem cell transplant conditioning. So there's not a lot of, I guess, methodical studies that have been done into the reasons and the rationale for lymphodepletion. It does work, but it does provide a number of undesirable features, including peri-infusion toxicity, immunosuppression, and so on, and obviously patient selection bias. So I would consider lymphodepletion, in fact, a handicap, but it was a conventional wisdom and still is in many cases that it is required for adoptive cellular therapy. Now I'm just going to dispense this very quickly by saying that it's not required, and in fact, studies have shown that if you use memory type T cells that we have, you can see long-term persistence of these cells for months or years, and that for the first time, actually, we showed in a Science Immunology paper that the clonal half-life of these T cells correlates with clinical response, and none of these patients received high-dose lymphodepletion. And I think that is a characteristic of preserving the natural circuitry and the intact cues. So let's take a look at the next bit of conventionalism, which has been very popular for the last 5 to 10 years. And that is the only good antigens are neoantigens. Now, I don't think everyone believes this, but it is certainly been riding high and very quickly over, let's say, since 2008 in the form of personalized neoantigens. And this is a sort of a timeline, sort of a vague representation of the approximate discovery, as well as the prevalence of particular TCR antigen in terms of coverage of patients and tumors. So because personalized neoantigens are individualized, it sort of sits here on the bottom, really private antigens, for example. But certainly neoantigens can take the form of virus-associated antigens, that HPV in head and neck cancer and cervical cancer, EBV-specific cells, T antigen, polyoma T antigen in Merkel cell, which we have targeted with cell-based therapies, for example, or HTLV1 and ATLL. And the other types, obviously, are an increasing interest in shared neoantigens, such as those represented by mutated KRAS, TP53, and EGFR, and answers the call for broad applicability. But let's take a step back and ask,
why are we looking at TCR targets in discussion? Why not CAR T cells? Don't we have enough targets there? And the answer to that is we do, but not really. I guess you could say that CAR T cells, which target the surface of a tumor, represent targets on the peel of the apple. We all know CAR T cell targets set the stage with the work of Michel Salin and Carl June. But if you look at the totality of target antigens for cell therapy, vaccine therapy, you see that perhaps TCR targets, which here represent the core of the apple, provide a much greater source of potential targets. Now, they are HLA restricted, but antigen loss is less likely, and you have thousands to choose from. And so just to remind everyone, basically the T cell receptor sees peptide MHC, but that this tumor antigen comes from any compartment of the tumor cell, whether it's endosomal, nuclear, transcriptional, surface, MDR genes, for example, but that protein must be internalized, processed, and presented on the surface as a peptide. So let's go back and say, were neoantigens the most immunogenic? And unfortunately, they're not. They are TCR targets, but we're not the first or even the most immunogenic TCR targets for immunotherapy. And for that, we have to go back to 1991, where Thierry Boone's group were the first to identify immunogenic TCR targets for melanoma and other cancers. By pulling out tumor-reactive T cells, it's able to screen tumor cDNA libraries and identify for the first time human target antigens and its derived immunogenic epitope that was recognized by human T cells through its T cell receptor and was able to reject tumor. So the first antigen was MAGE-1, melanoma antigen 1. He also identified melon A and then tyrosinase. Later on, Rosenberg's group identified and then rebranded melon A as MART1 for some reason, and then GP100 by Dr. Kawakami and others. And then Lloyd Old's group identified NY-ESO1, both class 1 and class 2 restricted epitopes, and certainly there's been a whole family of CT antigens which are found to be potentially immunogenic since that time. PRAME and MAGE-A4 being the most prominent of those, and now in clinical trials. Just to remind everyone, these are non-mutated tumor-associated antigens found in the cytoplasm or the nucleus and presented to TCR and are highly immunogenic. So what's old is new again. This is a quote by Winston Churchill, no Mark Twain, no Jonathan Swift, no Publius, actually Ovid. So really, recycling an old quote really is sort of pathognomonic of what we're doing here, is recycling the concept of immunogenicity. So let's try and understand immunogenicity a little more, because that is the underlying basis for TCR-based therapies. And neoantigens, by and large, have been identified by in silico identification of potential mutated antigenic epitopes. But is that considered immunogenic? Because for something to be immunogenic, you have to show that that target epitope was presented by the tumor cell in the context of MHC. You have to ask, does that peptide C and peptide MHC complex elicit a T cell response? And does that T cell response exhibit, or that TCR, sufficient affinity or avidity to recognize the tumor cells? And finally, if it does, is the peptide MHC complex presenting the epitope of sufficient density to be recognized? And then can you transfer findings at T cell receptor specificity to other non-specific T cells? And so if you look at the conventional approach of in silico prediction for neoantigen discovery, it does not answer any of these criteria for immunogenicity, at least not alone, not without some empiric validation. Unfortunately, tens of billions of dollars and tens of thousands of patients have been invested in this effort with some success, but generally very, very modest outcome, and in part because a lot of the epitopes that have been identified or proposed for neoantigen immunotherapy are not immunogenic. And I think that's really very important when people get on sort of the landslide of perhaps the allure of AI or large language models or computational biology, that this needs to be empirically validated. And so to do this, we sort of give you a little bit of history of neoantigen discovery. And there was a paper published in 2008 that suggested there may be neoepitopes in breast and colorectal cancer arising from known mutations. As well as that, there was a huge number of trials targeting neoantigens predicted neoepitopes. And so one of the premier groups in the field of mass spec and tumor antigen identification coming out of Germany took a closer look at this. And so they identified by in silico analysis about 1,300 neoepitope candidates. This is in a mouse tumor line, MC38. The random mass spec, only 7 of them were actually processed and presented with sufficient abundance to be recognized on mass spec. So that's 0.5% of them. And only 1 of these 7 actually proved to be immunogenic. So a fairly small fraction of the predicted candidates. And in 2020, the Parker Institute sponsored a trial and invited 25 teams, both academic and industry, to submit their candidate epitopes. They were all given the same exome sequencing data from 6 different tumor samples. And guess what they found? Anywhere from 7 to 82,000 peptide MHC candidates. So certainly not a consensus by any means. Nevertheless, they took the top 608, which is 0.7% of these, and then further refined this to the top 51, which were tested for immunogenicity. Only 6% of them were immunogenic by the loosest definition of immunogenicity, which was throw the peptides in, see if they reacted, or generated some T cell response, which doesn't actually speak to whether that antigen or epitope was processed or presented where it can induce any of the T cell responses or met any of the criteria, but only 6%. And so it's a vanishingly small number of predicted epitopes that are in fact immunogenic. Not that there aren't some that are present, but these are relatively small to begin with. So if we collapse this and take a closer look at how do you actually find these immunogenic epitopes and targets, you have to go old school. You have to do the difficult work, actually, of growing up hundreds of millions of cells, looting off the peptide off affinity-bound MHC, class 1 and class 2, and then running it through the mass spec. And this is a bit of an art as well as a science. I'm very fortunate that Pan and Yulin Xu in the lab have really dedicated the last 7 years of their life to looking at this and have generated a fair number of spectra, reduced that and deconvoluted to 6,500 targets, of which we curated 250 epitopes. So it's okay to apply AI and computational biology methods, but only after you have the empiric data. And this is certainly not our work alone. This is other work by Tony Weinschenk, Hanshul Ramansi, Ben Gungelhard, and others. And I think what our approach really has been is to extend that by synthesizing these particular peptide epitopes and using the ETC workflow, which I showed you before, can actually isolate very, very low frequency T cells from the peripheral blood to validate this in a way that's basically satisfied the criteria. Does this epitope elicit a T cell response? And does this T cell exhibit sufficient affinity to recognize tumor cells? And once you prove that, then you can go to the next step and prove, in fact, that you can transfer the T cell receptor specificity and completely validate these epitopes identified in the first place through mass spec. So what did we find? This is a TCGA database on the left in yellow, and then also the GTEx expression of RNA data or transcriptional data. I'm sorry the print is so small, but you can imagine this is really the profile of all the different tumor types, ranging from leukemia to uterine sarcomas to melanoma to prostate bladder cancer and so on across the top. And the reason I'm giving you this is sort of a gestalt of what is already known. And you can see from here, perhaps faintly, that the melanocyte-associated antigens and sort of this heat map considered tissue-specific as well epitopes for NY-ESO and MAGE-A4 represented here are really what's available today, primarily HLA-A2 and obviously some normal tissue expression in the testis for the CT antigens, which captures really only 3% of all cancer patients, not just those who are eligible by expressing these antigens, but really a very small fraction of patients. And what we discovered using this immunopeptidome database that Yulin and Kay have been working on is actually much larger, a more significant, highly expressed set of immunogenic targets. So you can see, for example, this is really a heat map. Everything above 4 transcript per megabase is considered potentially immunogenic, processed and presented. Here we're in the tens to hundreds compared to what's been discovered and tested right now in clinic, which is basically 2 to 3 transcript per megabase. So all these epitopes here belong to one antigen. In this case, some of these are cancer testis antigens, but they're all different epitopes presented by different alleles. And you can see they cover a very broad number of potential tumors. We curate them so they're all either germinal tissue restricted or tissue restricted. And so there are a lot of tissue specific targets melanocyte, prostate, and breast. I'll touch on that a little bit. You can see right here for this particular set of epitopes belonging to an antigen which is highly expressed in prostate cancer, for example, is also expressed in normal prostate tissue. And then surprisingly, there were a number of epitopes which came from antigens associated with cell cycle proteins, the kinetic core complex, DNA replication, DNA repair, of course, which are important, especially in chemo or targeted resistant tumors. And I think that this is actually quite interesting. You would think that there would be very little immunogenicity against sort of these essential proteins, but they are, and they're present at very, very high levels. We believe that there's a therapeutic window in which you can treat patients because those tumors express these consistently and very high levels. And certainly for the DNA repair genes, as well as a number of resistance genes, such as the MDR gene, which epitopes have been identified across a number of different alleles, representing about 80% of all patients. This is this class 1. In fact, Yulin took this to an even larger scale, and with the collaboration with CARPAN and also investigators McCoss and Wang at University of Washington, has compiled this database of shared tumor-associated repository, or STAR. Everything needs an acronym. This is our acronym, but it is one of the largest nonprofit academic tumor immunopeptidome databases for TCR therapies. And I'm not going to go through this poster, obviously, and I can send you a reference if you like, but the amount of work that went into this in order to de novo perform the analysis on 30 plus tumor lines and samples is actually heroic. We contributed more than 2 million novel spectra to a total of 20 million spectra to what's available, and he curated through the public domain a total of 180 tumor samples, 200,000 targets, several pan-cancer antigens, several that are tissue-specific, and as I mentioned too, associated with tumorigenicity, and also rare cancers as well, with global coverage of more than 80% of HLA types. We'll make this available through the program T-cell-based therapeutics, the STRIDE program sponsored by the Office of the Chief Scientific Officer, but this is one of the things that sort of is a linchpin moving this forward in the patient-centric tumor agnostic approach. And so, for example, here are some of the pan-cancer antigens which are expressed with 200,000-fold higher than in normal tissues. We've seen this particularly in glioblastoma and anaplastic thyroid cancer, and certainly multiple disease indications in solid liquid tumors. This one particular target actually is kinetic core complex protein presented by several different alleles, and you generate the T cells, as I've shown you in the schematic previously, so and expand them. They kill pancreatic cancer cells very well. And for illustrative purposes, they're relatively low affinity for GBM, but if you transfer to TCRT, you have fairly good killing of both the pancreatic, obviously, and the glioblastoma tumor at very low E-to-T ratios. You're looking at 5 to 1 here, 2.5 to 1, and so on. Normally, the E-to-T ratio is used to 40 to 1 or 20 to 1. And then certainly one of the things I like to focus on is anaplastic thyroid cancer, because we've done a lot of work in this area in collaboration with Stephen Lai and his team. Let me go through the rest of this, and I'll focus a little bit on ATC in a minute. So here, as I promised you, the targets for prostate cancer, highly expressed in prostate tumors, and certainly in normal prostate, considered a dispensable organ. Leukemias, lymphomas, very highly expressed. We have a gametocyte-specific transcription factor, which Casey Burmack in the lab is working on, highly expressed in testis and general tissues as well, but also found in T-cell lymphomas and T-cell leukemias, B-cell lymphomas, leukemias as well, and liver cancers. Breast cancer, sorry, I had to redact some of this, but one of the targets is mammoglobin, which is a biomarker for prognosis in breast cancer, found in triple-negative breast cancer, as well as anchorin is NYBR1, and highly expressed in breast cancer, anything above 3 or 4, as I mentioned to you, is immunogenic. And YEE, so for example, is TPM of 2.5. So these are highly immunogenic, potentially highly immunogenic, and in this particular case, mammoglobin is expressed in breast tissue, but not any other normal tissue. It makes it a fairly good target for both late stage and potentially early stage breast cancer. What about rare cancers? So, as it turns out, as long as the antigen is shared or expressed, the xenonon-mutated proteins, it turns out the epitope is also shared if a corresponding tumor or even tumor of different tissue type expresses that HLA allele. So a bit surprising, but that's basic biology in terms of antigen presentation, processing presentation. So that if either RNA-seq or proteomics data from cell lines, organoids, or anything In this case, let's pick rare cancers as an example. When you run the RNA-seq through the immunopeptidome database, you actually come up with a number of feasible targets, some of which we've already generated T cells to, and in collaboration with Al Young and Fred Lang in neuro-oncology and neurosurgery, Bernard Ravi in sarcoma, Tony Conley with chordomas, and Stephen Lyons, I mentioned, with ATC. Several of these T cells that we've already isolated, TCRTs, do kill these either glioblastoma stem cells or these other tumor cell lines that have been established here in the Anderson. So a very robust method, doesn't require us to grow hundreds of millions of cells, but really just the RNA-seq data. And so here's a target which has been described for years, PBK. It's a kinase, and it's associated with invasiveness, EMT transformation. It is actually considered a classic cancer testis antigen. So largely expressed only in germinal tissues, not talked about much because it's not one that we're familiar with seeing. It's not a major NY-ESO, but it is tumorigenic, and as it turns out, very immunogenic. And so the T cells that we isolate from this using the ETC protocol, from which we then identify the TCRTs and transfer them to lymphocytes, can kill anaplastic thyroid cancer, even at very, very low ETT ratios. Again, I'm showing you ETT ratios here, less than 10 to 1. For all these anaplastic thyroid cancer lines established in Stephen's lab, Ying Henderson, to give a shout out to for helping us grow all this. Paul Hung is the graduate student who's been doing all the work, and we have a manuscript in preparation showing that they killed this in the antigen-specific manner. And then actually the key to a lot of antigen discovery, epitope discovery with T-cell-based therapies is to prove in fact that the T cell kills the tumor by T cell recognition of the peptide MHC. And there's a lot of bystander killing, as you know, T cells and K cells, what have you. They can make granzymes, perfluin, fast-like interaction. Other TNF family members can do a lot of bystander killing. In fact, Bill Saxena, who's in the lab, but who was in the lab, is now with Gilead, has done a number of studies while in the lab looking at bystander killing. And really the results are profound. I can't discuss them right now, but there's a huge effect of non-TCR mediated bystander killing. And to prove, in fact, that PBK epitope is what is mediating killing and that the TCR is specific, you do a co-target inhibition assay where you throw in the co-target to block TCR engagement of the hot target, which expresses the epitope. And you see here that with all the necessary controls, this is clearly killing that's mediated through T-cell recognition of the PBK-specific epitope. But it turns out that PBK not only is found in rare cancers, but also in common cancers. And this is a chart over here showing its expression between 4 and 10 transcript megabase and greater than 10 across a number of different tumors, solid tumors, in which there's clearly an unmet need. Again, sorry, I just throw this up here to show you that a lot of work has been done showing it's highly expressed in prostate cancer. Pretty good H scores in ovarian cancer, for example, and certainly associated with poor prognosis in lung cancer as well and some of these other cancers. We also found recently that it can kill glioblastoma stem cell lines very well. So certainly a lot to be said for this sort of new, old panel of tumor-associated antigens, which were probably there, but not identifiable until recent technology became available to us. And also a lot of people doing the hard work. And so what we've got here really is a new player, really an old player in a new town. And I think this really speaks to how you may want to take a closer look at what's right in front of you. I'm going to mention also that we've identified possibly a transposon-associated epitope associated with a RNA virus. So what's old is new again. Certainly, or what's new is new again, I guess you could say that. So we don't think that the only good antigens, neoantigens, although they certainly play an important role, we should look elsewhere as well. And then the final thing I want to talk about in the last 6 to 7 minutes here is that single patient INDs are to be avoided. And I understand that, you know, especially at a large institution like MD Anderson, you have the resources, patient population, experts like David Hong and Van Merrick to engage groundbreaking studies and change the standard of care with large-scale studies. But I really would like to point out that in the area of grassroots innovation, single-patient INDs are, in fact, very often where it all starts. And so let me give you an example. The power of one, I guess is what I like to call this. And so here's the case of Elsie. Elsie was a very public figure who started Stand Up to Cancer, but herself was a patient with metastatic breast cancer. In 1999, as I mentioned to you a long time ago,
Tetramer-guided cell sorting strategy was developed in our lab and published and showed that you could, in fact, isolate and expand tumor-reactive T cells from the peripheral blood. This was all in the lab, though. And later on in 2005, 2008, and then in 2020, where we defined the mechanism in which this occurs, adding ALK21 priming actually gave us a nice population of central memory T cells, which then coupled with tetramer-guided cell sorting allowed us to isolate and expand any specific T cells in a manner that was relatively reliable and routine for immunogenic epitopes. And so somewhere during this time, so now we're looking at 10 years after tetramer-guided cell sorting and 5 years after ALK21 discovery, we get a call from Tony Ribas in LA about patient LZ who's failed multiple lines of therapy, including autologous stem cell transplant, which was in the vogue at the time for breast cancer. We met with SP, her friend at Center for Cancer. Turns out she has an NY-ESO positive tumor and potentially an A24 restricted epitope, which no one had grown T cells to before, and we had neither. But we went ahead with the leukapheresis anyway. We could not apply the cell sorting technology or the ALK21 priming because we didn't have a GMP grade cell sorter, wasn't FDA approved. We just decided we'd go ahead and do brute force cloning and clone out 10,000 clones and see what we can get. The next month, check arrives in the mail from SP to purchase a grade cell sorter, causes a lot of commotion, but we get the cell sorter. And we submit a single patient IND, or a CIND as called at that time, to the FDA for first in class use of a cell sorter, first in human use of ALK21 prime tetramer-guided T cells. Now you couldn't do this with an IND. It would be impossible. By this time, also the opportunity would have passed. There was not a whole lot of engineering data, but I think that this was reached a sufficient threshold that the FDA felt was safe. Certainly, there's an imminent clinical need. The sorter arrives in January, and it doesn't work. And basically, it's because this little plastic spigot needs to be carved out by hand in order to connect it from the sheath tank through the sorter right to the sorting arena. And I went down to Stanford and they showed me how to shave off this spigot, and they were using this to isolate regulatory cells in Rob Negrin's lab, but nothing for tetramer-guided cell sorting. And so literally a few weeks later, FDA grants the CIND or SPIND, and we treat this patient. So literally a month from when the sorter arrives, we could get all this in place that we waited more than 10 years for. We treated with NY-ESO specific CD. What do we see? So it turns out when you infuse these NY-ESO specific T cells, you can see them. No conditioning or very low conditioning, low dose conditioning. They were present in 1% of all CD8 T cells, which is not a bad amount, 3, 4, 2 weeks later. But we wanted to give her a boost, so we gave it again. Now these cells are there, again, 1%. But we had to send it back. So they may be present much lower frequency for longer term, but we sent it back to Tony Ribas and John Glaspie. And at that time, he had access to one form of anti-CTLA-4. And we gave that to the patient without any further infusions. It turns out these cells were being suppressed by some regulatory mechanism, and bang, 3.5% of peripheral circulating T cells were present. We were able to enable first-in-class use of a self-contained nanosorter for use under GMP conditions, and able to add immune checkpoint inhibitor for the first time to any cell-based therapies. So what did we learn from this one patient? Well, we were able to take a protocol that had taken us 12 weeks to generate T cells down to 7 weeks, which is not great. We're trying to move that further down to 4 to 5 weeks, and certainly spawn off the TCRTs with TCRT-based therapies, but certainly enabled a number of things. Tetramer-guided cell sorting was established from this one patient, T-cell memory reprogramming, combination of CTLA-4, elimination of conditioning lymphodepletion, and induction of antigen spreading. And because of this one patient, the FDA allowed us to apply for an IND, for which we received approval to treat other patients. Allowed us to cure this patient, who had a high volume disease, allowed us to cure this other patient. This is Brian. He was featured on Stand Up to Cancer, American Airlines pilot who actually had a brain met. And under the care of Hussein Tabi, referred it to our program, and he's essentially cured now. On the 5th year anniversary of his complete response, he sent me this airplane from American Airlines. He had just gotten his recovered or retaken or reapproved for his American Airlines commercial pilot's license. And so this is the best gift I've ever gotten from any patient. It spawned a number of clinical trials. I'm not going to go through them all, but certainly dating back over the last 10 years, 11 years, not just for anaplastic thyroid cancer and melanoma, but also hopefully a trial with Dr. Glitzer using intrathecal ETC with leptomeningeal disease. And then what about where it really counts? Funding, $25 million in NIH funding as a result of this one patient, which led to IND approval with these several trials, $8 million in CPRIT funding, $22 million in philanthropy at the Fred Hutch, where this first patient was treated, $2 million in philanthropy at MD Anderson, 18 issued patents, first-in-class GMP self-sorter technology license. And this technology, the CTC technology, which is all encompassed and was used in this patient, was licensed to a company which IPO'd a $2 billion cap, also started another T-cell therapy company. And then, as you may have heard, NASA sponsored the first T-cells in space grant here at MD Anderson. We're fortunate to receive that last year. And so I think I've talked a little bit about how really don't really like to follow conventional wisdom sometimes, why it's important, to question long-held beliefs by asking simple questions. Not that conventional wisdom is important, but once in a while, just take a look and take the shine off some of these. And rather than avoiding single patient trials, it should be encouraged. If you have a lot of the technologies and the dedication to try and understand how things work, not treating them off as a one-off, in this case, spawn a number of different trials and technologies, which have been universally helpful. So the ultimate goal of any treatment modality is to develop a patient-centric tumor-agnostic approach. And the linchpin for this, at least in our case, was an immunopeptidome data set, which we believe is immunogenic, is improved in more than 24 different epitopes we've formally validated. And then to define a niche in which this new modality plays in. And so certainly with all these targets, we can cover and potentially treat a number of common and rare tumors where these antigens and epitopes have been identified. Because we're unbiased in HLA selection and how we identify these, covers more than 80% of the world population based on classical alleles. And this is the immunogenicity criteria, which we think is extremely important, which fortunately we have a handle on based upon 20 years of work in ETC-based therapies. And what's the application of all this? Well, you can take it and you can do endogenous T-cell therapy, obviously, as we've been doing, or you can take the TCRs, which have been validated, and put them into TCR-T and KR and your effector of choice. TCR mimics against a lot of these newly defined peptide MHCs for bispecific engagers, and then obviously vaccines, of which there's many, many ways to deliver them. And then the one thing we're doing with Lin-Hua is looking at T-cell cooperative response as well, because now we have a very, very large database of T-cell receptors and we know what they recognize. What about endogenous T-cell therapy? It does have a number of advantages: memory, low toxicity, no conditioning, agile targeting. You just need GMP-grade peptide and tetramer. What's the niche? Perhaps minimal residual disease, early stage disease from colorectal cancer, ovarian, GBM, or breast cancer, in discussions with Scott Kopetz and others to look at this. Perhaps a huge unmet need, cancer unknown primary. We treat on the basis of histology, give it a 5FU if it looks like it's glandular, or if we're lucky, maybe it's a lymphoma. But I think basically, if you know the RNA signal tumor is truly patient-centric and tumor agnostic, we may have a target for already, for which we may already have a TCR-T. Leptomeningeal disease, as discussed already. And then, of course, because of the low toxicity and the possibility of combination-based therapies, you have all these other strategies now combined into looking at this as a patient-centric tumor-agnostic approach. And that is the end of my talk. I just want to thank all the people in the lab here, some of which are highlighted: Kirpan, Ivy Lai, who has been responsible really for moving all this into the clinic in the GMP facilities, both at P14 and elsewhere. Yating Li, graduate student Jimmy Wong did a lot of studies with all-21. Priscilla Ortiz, who helps run the lab. Shabala Singh, who basically does everything in the lab, is principal scientist and general lab manager. Yulin, I mentioned about computational biology. Farah, who's involved in studying tissue-resident memory cells. Our collaborators in rare cancer world. Osha Pui and Seth Polak, former postdocs, now seasoned and veteran faculty members in very nice positions, continuing to work at T-cell-based therapies. Melinda Mata, a scientific director whom we hired a year and a half ago for the program T-cell-based therapeutics, coming out of the CSO office, really somebody to contact if you wish to collaborate. So this is a bit of an advertisement for that. And then funding for all these sources.